In this video, I'm going to talk about cell communication, the cell cycle, aka mitosis, or including mitosis, and cell cycle regulation. So in cell communication, we learned about a couple different types of cell communication. We learned about cell-to-cell communication. That includes autocrine, which is the cell sending signals to itself, dextocrine, which is cells that are right next to each other with a receptor and a ligand attached to them, touching each other. Short distance cell communication, like paracrine, where one cell releases a signal that then is received by another cell, so short distance. And long distance, like endocrine, where cells from one part of our body send a signal that's received by another cell in another part of our body. In all cases, when the ligand binds with the receptor on a cell, we call that reception. And that triggers transduction in the cell, which often involves amplification and a phosphorylation cascade. So you may remember one protein might phosphorylate another protein, and this one protein can phosphorylate many, and then these phosphorylated proteins can phosphorylate many of the next kind. And those phosphorylated proteins carry out the action that is dictated by that cell communication. So, for example, some responses are maybe it could turn on or off a section of DNA, so increase or decrease the production of a protein. It could activate an enzyme or deactivate an enzyme, or it could trigger apoptosis in the cell. It could also have some effect on the cell cycle, on the cell doing mitosis. So let's talk about the cell cycle. So the cell cycle includes interphase and mitosis. So most of the time cells are in interphase, only a little bit of the time they're in mitosis. So you may remember there's G1 phase, S phase, G2 phase, and M phase. G1, S, and G2 are all part of interphase. G1 and G2 both involve growth. S stands for DNA synthesis. So this is when the DNA is copied. Mitosis is when the cell actually does the splitting. You'll also notice these little flags. That's a reminder that there's checkpoints in the cell cycle, and we're going to talk about that in a second. And G0, remember, is if the checkpoint at the end of G1 decides that the cell isn't ready to go to S phase, then the cell gets stuck in what we call G0, like nerve cells that aren't going to divide are in G0 phase. Mitosis, as a reminder, just because this came up in a lot of your FRQs, there are 4 main phases of mitosis. Prophase is when the DNA condenses so that it's manageable. Metaphase is when the chromosomes line up in the center. And anaphase is when the sister chromatids are pulled apart to either sides of the cell. Telophase and cytokinesis is when the cell starts to actually split into two. So these green lines are microtubules that extend out and attach to the chromosomes. And as a reminder, there is a motor protein that grabs onto that chromosome and walks on the microtubules to pull the chromosome back to either pore. That's how that works. Okay, cell cycle checkpoints. So we learned in a POGIL that cell cycle checkpoints are regulated by a combination of cyclin-dependent kinase, CDK, and cyclin. So when these two molecules come together, we call them a maturation promoting factor, MPF. They also need a phosphate group. And these two molecules have to come together to be able to signal the cell that it can go past the checkpoint and on to the next phase. So we can see here that in this example, cyclin-dependent kinase, CDK, is always present, but cyclin cycles and is sometimes high and sometimes low. And you'll notice here, this must be for the cell cycle checkpoint that allows the cell to go into mitosis, because when the cyclin is high, and the combination of the two is high, that's when the cell's moving into mitosis. So remember, CDK and cyclin control the cell cycle checkpoints. And we need regulation of the cell cycle to prevent cancer so that cells aren't just dividing willy-nilly.